Endoplasmic reticulum.
The endoplasmic reticulum is a type of organelle in the cells of eukaryotic organisms that forms an interconnected network of flattened, membrane-enclosed sacs or tubes known as cisternae. The membranes of the ER are continuous with the outer membrane of the nuclear envelope. Endoplasmic reticulum occurs in most types of eukaryotic cells, including the most primitive Giardia, but is absent from red blood cells and spermatozoa. There are two types of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. The outer face of the rough endoplasmic reticulum is studded with ribosomes that are the sites of protein synthesis. The rough endoplasmic reticulum is especially prominent in cells such as hepatocytes where active protein synthesis occurs. The smooth endoplasmic reticulum lacks ribosomes and functions in lipid metabolism, carbohydrate metabolism, and detoxification and is especially abundant in mammalian liver and gonad cells. The lacy membranes of the endoplasmic reticulum were first seen in 1945 by Keith R. Porter, Albert Claude, Brodie Meskers and Ernest F. Fulham.
Structure.
The general structure of the endoplasmic reticulum is a membranous network of cisternae, sac-like structures, held together by the cytoskeleton. The phospholipid membrane encloses a space, the cisternal space, or lumen, which is continuous with the perinuclear space but separate from the cytosol. The functions of the endoplasmic reticulum can be summarized as the synthesis and export of proteins and membrane lipids, but varies between ER and cell type and cell function. The quantity of ER and SER in a cell can slowly interchange from one type to the other, depending on the changing metabolic activities of the cell. Transformation can include embedding of new proteins in membrane as well as structural changes. Changes in protein content may occur without noticeable structural changes.
Rough endoplasmic reticulum.
The surface of the rough endoplasmic reticulum, often abbreviated RER, is studded with protein manufacturing ribosomes giving it a rough appearance, hence its name. The binding site of the ribosome on the rough endoplasmic reticulum is the translocon. However, the ribosomes bound to it at any one time are not a stable part of this organelle's structure as they are constantly being bound and released from the membrane. A ribosome only binds to the RER once a specific protein nucleic acid complex forms in the cytosol. This special complex forms when a free ribosome begins translating the mRNA of a protein destined for the secretory pathway. The first 5 to 30 amino acids polymerized encode a signal peptide, a molecular message that is recognized and bound by a signal recognition particle, SRP. Translation pauses and the ribosome complex binds to the RER translocon where translation continues with the nascent protein forming into the RER lumen and/or membrane. The protein is processed in the ER lumen by an enzyme, a signal peptidase, which removes the signal peptide. Ribosomes at this point may be released back into the cytosol. However, non-translating ribosomes are also known to stay associated with translocons. The membrane of the rough endoplasmic reticulum forms large double membrane sheets that are located near, and continuous with, the outer layer of the nuclear envelope. Although there is no continuous membrane between the endoplasmic reticulum and the Golgi apparatus, membrane-bound vesicles shuttle proteins between these two compartments. Vesicles are surrounded by coating proteins called COPI and COPII. COPII targets vesicles to the Golgi apparatus and COPI marks them to be brought back to the rough endoplasmic reticulum. The rough endoplasmic reticulum works in concert with the Golgi complex to target new proteins to their proper destinations. A second method of transport out of the endoplasmic reticulum involves areas called membrane contact sites, where the membranes of the endoplasmic reticulum and other organelles are held closely together, allowing the transfer of lipids and other small molecules. The rough endoplasmic reticulum is key in multiple functions. Manufacture of lysosomal enzymes with a mannose 6-phosphate marker added in the Golgi network. Manufacture of secreted proteins either secreted constitutively with no tag or secreted in a regulatory manner involving clathrin and paired basic amino acids in the signal peptide, integral membrane proteins that stay embedded in the membrane as vesicles exit and bind to new membranes. RAB proteins are key in targeting the membrane. SNAP and SNARE proteins are key in the fusion event, initial glycosylation as assembly continues. This is N-linked, O-linking occurs in the Golgi. Unlinked glycosylation, if the protein is properly folded, glycosyl transferase recognizes the AA sequence NXS or NXT, with the ST residue phosphorylated, and adds a 14-sugar backbone, 2-N-acetyl-glucosamine, 9-branching mannose, and 3-glucose at the end, to the side chain nitrogen of ASN. Unlinked glycosylation, if the protein is properly folded, glycosyl transferase recognizes the AA sequence NXS or NXT, with the ST residue phosphorylated, and adds a 14-sugar backbone, 2-N-acetyl-glucosamine, 9-branching mannose, and 3-glucose at the end, to the side chain nitrogen of ASN.
Smooth endoplasmic reticulum.
The smooth endoplasmic reticulum, abbreviated SER, has functions in several metabolic processes. It synthesizes lipids, phospholipids, and steroids. Cells which secrete these products, such as those in the testes, ovaries, and skin oil glands, have a great deal of smooth endoplasmic reticulum. It also carries out the metabolism of carbohydrates, drug detoxification, attachment of receptors on cell membrane proteins, and steroid metabolism. In muscle cells, it regulates calcium ion concentration. It is connected to the nuclear envelope. Smooth endoplasmic reticulum is found in a variety of cell types, both animal and plant, and it serves different functions in each. The smooth endoplasmic reticulum also contains the enzyme glucose 6-phosphatase, which converts glucose 6-phosphate to glucose, a step in gluconeogenesis. It consists of tubules that are located near the cell periphery. These tubes sometimes branch, forming a network that is reticular in appearance. In some cells, there are dilated areas like the sacs of rough endoplasmic reticulum. The network of smooth endoplasmic reticulum allows for an increased surface area to be devoted to the action or storage of key enzymes and the products of these enzymes.
Sarcoplasmic reticulum.
The sarcoplasmic reticulum, SR, from the Greek sarx, flesh, is smooth ER found in smooth and striated muscle. The only structural difference between this organelle and the smooth endoplasmic reticulum is the medley of proteins they have, both bound to their membranes and drifting within the confines of their lumens. This fundamental difference is indicative of their functions. The endoplasmic reticulum synthesizes molecules, while the sarcoplasmic reticulum stores and pumps calcium ions. The sarcoplasmic reticulum contains large stores of calcium, which it sequesters and then releases when the muscle cell is stimulated. It plays a major role in excitation-contraction coupling.
Functions.
The endoplasmic reticulum serves many general functions, including the folding of protein molecules in sacs called cisternae and the transport of synthesized proteins in vesicles to the Golgi apparatus. Correct folding of newly made proteins is made possible by several endoplasmic reticulum chaperone proteins, including protein disulfide isomerase, PDI, ERP29, the HSP70 family member BPGRP78, canexin, calreticulin, and the peptidylpropyl isomerase family. Only properly folded proteins are transported from the rough ER to the Golgi apparatus. Disturbances in redox regulation, calcium regulation, glucose deprivation, and viral infection or the overexpression of proteins can lead to endoplasmic reticulum stress response, ER stress, a state in which the folding of proteins slows, leading to an increase in unfolded proteins. This stress is emerging as a potential cause of damage in hypoxia, ischemia, insulin resistance, and other disorders.
Protein transport.
Secretory proteins, mostly glycoproteins, are moved across the endoplasmic reticulum membrane. Proteins that are transported by the endoplasmic reticulum throughout the cell are marked with an address tag called a signal sequence. The N-terminus, one end, of a polypeptide chain, that is, a protein, contains a few amino acids that work as an address tag, which are removed when the polypeptide reaches its destination. Proteins that are destined for places outside the endoplasmic reticulum are packed into transport vesicles and moved along the cytoskeleton toward their destination. In human fibroblasts, the ER is always co-distributed with microtubules, and the depolymerization of the latter causes its co-aggregation with mitochondria, which are also associated with the ER. The endoplasmic reticulum is also part of a protein sorting pathway. It is, in essence, the transportation system of the eukaryotic cell. The majority of its resident proteins are retained within it through a retention motif. This motif is composed of 4 amino acids at the end of the protein sequence. The most common retention sequence is KDEL, Lisasp-GLU-LR. However, variations of KDEL do occur, and other sequences can also give rise to endoplasmic reticulum retention. It is not known whether such variation can lead to sub-ER localizations. There are 3 KDEL receptors in mammalian cells, and they have a very high degree of sequence identity. The functional differences between these receptors remain to be established.
Endoplasmic reticulum.
The endoplasmic reticulum is a type of organelle in the cells of eukaryotic organisms that forms an interconnected network of flattened, membrane-enclosed sacs or tubes known as cisternae. The membranes of the ER are continuous with the outer membrane of the nuclear envelope. Endoplasmic reticulum occurs in most types of eukaryotic cells, including the most primitive Giardia, but is absent from red blood cells and spermatozoa. There are 2 types of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum. The outer face of the rough endosome, and detoxification and is especially abundant in mammalian liver and gonad cells. The lacy membranes of the endoplasmic reticulum were first seen in 1945 by Keith R. Porter, Albert Claude, Brodie Meskers and Ernest F. Fulham.
Structure.
The plasmic reticulum is studded with ribosomes that are the sites of protein synthesis. The rough endoplasmic reticulum is especially prominent in cells such as hepatocytes where active protein synthesis occurs. The smooth endoplasmic reticulum lacks ribosomes and functions in lipid metabolism, carbohydrate metabolism. The general structure of the endoplasmic reticulum is a membranous network of cisternae, sac-like structures, held together by the cytoskeleton. The phospholipid membrane encloses a space, the cisternal space, or lumen, which is continuous with the perinuclear space but separate from the cytosol. The function